Okay, this third video is going to start with a typical neutrophil. This is a phagocytic cell designed to engulf and destroy pathogen. And your book does a lovely job of combining half TEM microscopy pictures, here's the TEM microscopy pictures, with half cartoon. So this actually does occur. You don't have to take my word for it. You can see that this cell engulfs bacterium, bringing it in. But let's look at the cartoon. This is a phagocytic cell, so it's going to engulf the pathogen. This membrane-bound vesicle that it brings it in is going to be called a phagosome. And the phagosome is going to merge with a lysosome. Remember in cell architecture lecture we were talking about how lysosomes are filled with digestive enzymes, toxic oxygen species, all kinds of nasty things that you don't want to be just running amok inside the cell. So when that lysosome meets the phagosome, you just smash those two words together, leads you to the phagolysosome. That's going to dump all of those digestive enzymes and all of that stuff onto the bacterium, which is going to kill it. And then waste will get spit out if there's any waste. So that's pretty impressive, but now we're talking about the epic battle of evolution, right? And how we've evolved to have our cells attack pathogen, engulf them, and destroy them. Meanwhile, organisms are not sitting idly by. Microbes are starting to evolve to be able to be engulfed by these macrophages and neutrophils, but before the lysosome meets with the phagosome, they escape. And then they are free to kind of run around the inside of that immune cell and hide from the rest of the immune system. That can be, really profound. It's a good way for these cells to hide out until they gain good numbers. I linked this video onto Blackboard. This video shows a typical cell, this is tissue culture cell, so not in an actual body, but they infected this cell with Listeria monocytogenes, the organism that causes listeriosis, and the cell engulfed it to bring it in, and all of those little black rods that are zooming around the cell are Listeria cells. They got out of the phagosome and they're just dinking around inside the cell, growing and replicating. This is the greatest microbiology video ever shot, I think. I dare you to watch it just once. You should watch it and then show it to all your friends and neighbors and see how cool the immune system is. This really and truly is the epic battle of good versus evil. I linked this one also onto Blackboard. This shows a white blood cell tracking down. This is real time too. This is basically, if you look at how this was done, they basically put a drop of blood in that was infected with a bacteria onto like a hanging drop mount, put it under a microscope, and then put a camera up to it. So these are live cells in real time, and you can see that the red blood cells are the circular type cells that don't do anything. The bacteria is a little rod bacteria that has tumbling motility, and the white blood cell is really and truly hunting it down and chasing it down to engulf it, to destroy it, to get it out of the system. So that's, a really big part of getting better, is to have those pathogens engulfed and physically destroyed by cells in your body. There are protein defense systems. Like I said, your immune system doesn't ever do anything halfway. It does everything at full tilt all the time. And there are a series of proteins that are in your blood called complement proteins. So complement is a system. It's a series of proteins that are in the blood that have a function on clearing pathogen. And there's a number of ways to activate these proteins or activate complement. We're only going to talk about one specific way to activate complement and some of the outcomes one gets when complement has been activated. But in this pathway, in the classical pathway of complement activation, you basically have these proteins, and one of the proteins, and of course they are all named and numbered, and you don't have to know the names or the numbers of the individual proteins themselves. That's a little bit more in depth than we need, but there's one of the complement proteins recognizes an antibody attached to a bacterial cell. An antibody is a protein made by the immune system. So there's a blood protein that recognizes that interaction and then binds to that bacterium, and then it's going to recruit its buddies, its other buddy proteins. That's amplification and cascade. And then all of those activated complement buddy proteins will all bind onto the bacterium, and they bind in a donut type configuration. The reason they bind in a donut type configuration is based on when they finally receive a signal, all of those proteins that are now bound to the bacterium inserts themselves into the bacterial cell wall. That's called the membrane attack complex, which is going to essentially mean a great big crater-sized hole in the bacterial cell. And if you can blow a hole into your bacterial cell, you're going to do a good job of killing it. So this slide is really busy, and there's a lot of words and numbers and things on it. And let me just give you the basic overall rundown. You have a pathogen. Looks like this particular one is a coccus-shaped bacteria, and it has antibodies bound to it. These red things are antibodies that are bound to that pathogen, meaning that the body already has that pathogen tagged for destruction. So now we have a complement protein that's circulating in the blood, is going to recognize that there's antibody bound to that pathogen, and that's going to activate it. And then it's going to get excited, and then it's going to recruit all of its buddies. So all of this down here is just basically saying that there's recruitment, there's amplification and cascade of all of these reactions with all of these proteins to get them really active. Then we end up with a bunch of proteins that bind to the pathogen in a donut type manner, and then upon a signal they all insert themselves in the membrane, making boom, blammo, they've poked giant holes. So this is a picture, a real close-up picture of what a membrane attack complex looks like, which basically is just a bunch of holes. This organism is Swiss cheese. It's not going to survive this at all. If this is the whole bacterial cell, I don't know, maybe that bacterial cell is somewhere in the neighborhood of 6 microns or something to that nature. Look how big each one of these holes is. Each one of those holes is very damaging. When you start having multiple holes like that, the cell's not going to be able to survive. So we are just talking about, and you're only responsible for the classical pathway in how these proteins become activated, but there are other ways to activate complement. So just in the back of your head, know that there are other ways that complement can be activated. Now, once complement is activated, all of these things happen. So it doesn't matter how complement becomes activated. Once it does, the outcome is the same. The membrane attack complex is this killing of pathogens, this direct whole blowing up of the pathogens. But there are some complement proteins that act as opsonins. Remember, that's a term that means a protein that sticks to a pathogen that's going to make it easier to phagocytose. So some of those complement proteins just stick to the bacterial cells, and other phagocytes that come through can go ahead and engulf them. Also, complement activation leads to a pro-inflammatory response. So you get inflammation, which means you're going to get more immune cells. So that's really, really helpful. So most of the discussions that we've been having on this video series is based on bacterial invaders. But you do get invaded by viral invaders too. And the mechanism of help can be a little bit different. Viruses are teeny, 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 tiny. It's very difficult for neutrophils and macrophages just to engulf them and destroy them. They're not bacterial. But you do have some home defense systems based on chemical signaling, and that is in the form of interferons, and specifically interferon alpha and interferon beta. These are chemicals that are made by cells that have already been infected by a virus. So if we look at, and again, this is a slide that has a lot of words and busy stuff, let me just kind of take you through it. Basically, you have a cell that's been infected by a virus, and that cell is doomed. But think about how the virus is going to proceed. It's got to recognize, attach, it gets into the host cell, then it starts to take over the host cell, then the host cell makes all the new variables. That takes time. So in the meantime, before the cell is completely just defunct, it's making interferon alpha and beta chemicals and secreting them out to neighboring cells. So those chemicals then are read by receptors on neighboring cells, and neighboring cells, basically it's like an SOS saying, I'm infected, I'm going down, everybody protect yourself. So what's going to happen is that these neighboring cells will start really ramping up their antiviral protections. Viruses have to recognize and attach to potential cells that they're going to invade. So one of the mechanisms of the interferon response is that these neighboring cells will kind of hide all their receptors. They cover their receptors with little caps to hide them from the virus. They also make certain enzymes that destroy RNA or DNA that's just floating around the cytoplasm. So if the virus does get into that neighboring cell, it's destroyed by these enzymes. So these are antiviral proteins that are secreted by doomed virally infected cells, but are read by neighboring cells so that they can protect themselves a little bit better. There is something called gamma interferon, though, and that's not antiviral. That is something that is made by T cells that's going to get certain phagocytes to really kill bacteria, and we'll talk a little bit about that later on when we talk about cellular immunity. So the last thing we want to talk about is a natural killer cell. Natural killers, I mean, it's a perfect name for them. They are natural killer cells. And what they do, and I link this video, is that they're circulating cells. And they basically are checkers. They're patrol cells. They come into contact with your cells, the rest of your cells, and they check your cells for healthfulness. If your cell is healthy and happy, nothing bad happens. But if your cell is infected with virus, these natural killer cells are going to check it, find that it's infected with a virus, and these natural killer cells kill it. So yes, one of your cells dies, but all of the viruses will die with it inside that doomed cell. So you have protection against viral invaders, but it works different because of the nature of viruses. It's different. So check out this video. It's pretty interesting.